So thank you, everyone, for coming. For those of you who don't know me, my name is Veera Baladanditapania. I'm the chair of biostatistics here at University of Michigan. So it's actually my honor and privilege to set up the stage for the keynote lecture here. It's a special lecture that we have set up in honor of Mike. And to give you a brief history of the award, this was established in 2022 by the Department of Biostatistics to honor Mike's legacy, for honoring excellence in biostatistics, statistical genetics, and genomics. And as you have heard, throughout this afternoon, and you'll hear more at dinner as well as tomorrow, Mike's had an impact at so many levels, scientifically, as well as, more importantly, the mentoring aspects of it. And when we were putting in these metrics together, it is just staggering numbers that what Mike's has achieved in terms of, and the impact is, palpable, especially in this room and all the remarks that have gone around, And his exceptional mentorship of guiding over 40 PhD students and postdocs and having 200 trainees touch points, and I'm sure many of you resonate with that. So this award was set up. It's selected annually by the biostatistics faculty, and then through internal nomination process. And this is the 4th year running for this award. Here are the past recipients. I know Nancy is here, who received this last year. Ken Lang was the inaugural winner back in 2022 with Sylvia Richardson, and Nancy won it last year. So this year's recipient, it's my distinct pleasure to welcome back Goncalo Abecasis, who's currently the Senior Vice President and Chief Genomics and Data Science Officer at Regeneron. But as you all know, Goncalo has a deep footprint in the department as well, was a faculty here for many years, and also served as the chair of Michigan Biostatistics, 2014 to 2018. So let's give a big round of applause for Goncalo. And now I'm going to hand it over to Dr. Jian Kang, the Associate Professor for Research, to introduce Goncalo and go through his accomplishments and in the context of this award.
Yes, thanks, Veera. It's my great pleasure to introduce Goncalo. So actually, I'm Jian Kang. I'm a professor, not an associate professor. I'm a professor and associate chair for research of biostatistics. And so Goncalo's career has been remarkably influential across the academic and industry leadership. So after earning his Doctor of Philosophy degree from Oxford, he came to Michigan and became one of the leading figures in statistical genetics. And along the way, he served as the department chair and led major genomics initiatives. He has contributed to projects like 1,000 Genome and PubMed, and his work has advanced GWAS genotype imputation, sequence analysis, and large-scale biobank science. And for those of you care about Google citation, I think Goncalo is a record in our department for the highest citation, like 400,000 citations. I think that's probably the summation of all other faculty in our department. Yeah, and it's really impressive. And so one of the remarkable thing about Goncalo's work that combines deep methodological innovations as well as a huge practical impact. His research helped make modern large-scale human genetics possible, from statistical methods for sequence analysis to tools for genotype imputation and complex trait analysis. And his work contributed to discoveries of diabetes, cardiovascular disease, and psychiatric disorders, and many other complex diseases. And he has played a major leadership roles in building genomic resources and collaborations that the entire field now depends on. So this award is especially meaningful because Goncalo's connection to Mike goes back to the beginning of his scientific career. And Mike recruited Goncalo to Michigan after hearing back his dissertation work. As Mike joked, it only took 2 minutes to conclude the master's. Sounds impossible, and a few more minutes to realize Goncalo had actually made it work. So their collaboration become one of the defining partnerships in statistical genetics, have shaped the modern approach to association studies, sequence analysis, population scale genomics. And more broadly, their story reflects what make Michigan biostatistics specialists a strong mentorship, bold ideas, and collaborations that truly move a field forward. So please join me in congratulating Goncalo as a 2026 Mike Boehnke Visiting Scholar Award recipient.
Pleased to be here, and and I think what made me happiest so far were a couple of conversations I had. One very brief with Mike, which was, you can't really be retiring. Are you really retiring? And he said, well, no, he's not really retiring, which was quite a relief to me. He said he loves what he does, and how could he do more fun stuff? And, I always worry that, people retire, then they don't have anything to do and they age faster. And that will be the cure to aging that Nick is trying to find. Just keep doing stuff you love. This photo from early on when I met Mike is very emblematic because, Mike was always very gracious in hosting us and many other colleagues at his home and at many different places. I think this photo is not actually at his home. It'll be challenging to figure out where it is, but not that challenging, And also behind Mike, his partner and really supporter, inspiration, Betsy, and they're all, the fact that they've been together so long and raised great kids is obviously a great inspiration as well. I thought I would try and, for this talk, to go and figure out Mike a little bit, and I thought, I should go read some of his early papers. Obviously, there's some that are really classics in the field, but I thought, maybe I should go read Mike's first paper. And I dug it out. But before I go there, I think if anybody wants to figure out a winning strategy to get to 400,000 citations, if you write about 600 or 700 papers, have every paper cite all the other ones, you'll be there. The hard thing, it's very easy to do it if you do the n times n minus 1 divided by 2 kind of number of papers. If you want to do the n squared, you have to figure out how to get the first paper to cite all the subsequent ones. But I think it can be done. And so Mike was not quite as methodical about kind of inflating citation counts, and so he's just behind me, but that's only luck. Now, this paper was not one of the classics in terms of number of citations, but I think I could find a few nuggets there that are very telling and really illustrate this point of how one's experiences shape us for the future. And so in this paper, Mike was struggling with his colleagues to quantify HLA frequencies, and this was with a very early kind of assay that I only vaguely understand, but I think
since been discarded.
So others might understand better. And so they had cell lines from the patients. They would culture them together with other cell lines that expressed the appropriate T cell receptors to co-stimulate each other, and they'd see what pairs of cells grew and you could decode HLA types. But there were a couple of comments that I thought were really quite relevant. And so at some point in the paper, very early on, they said, we've experienced wide experimental variations with the procedure despite repeated attempts to modify techniques or computer analysis programs. You know, some of the variability in the results is because the computer analysis programs were not very reliable. Now, one thing that I struggled to find, and I read through the entire paper because I was very curious, this is a 1980 paper, how are these computer analysis programs coded and run? Were they still on punch cards? Were they? But this really illustrates one of Mike's concern with getting to high quality science, to developing not just a quality statistical method, but a methodology and a package that would allow others to implement and use it. You know, and so because of these wide variations they were dealing with, they decided to maximize reproducibility. Now, the other comment, you know, when you think, you know, how Mike thinks about complex traits and the contribution of genes and the environment and multiple genes, this is maybe one of the first instances where he expressed that thought in press, at least. You know, at least as far as I could tell, it's his first paper, so it can't be far off. It say, may be influenced by a variety of immunological and non-hereditary factors and probably reflect the products of more than one genetic locus. You know. Now, I have to say that, you know, as you all know, Mike, as we know him now, is an extremely, extremely careful editor. And the typo on hereditary factors is not mine. It's in the printed paper. So, and I think a comma before despite wouldn't go bad either, you know. It's just a long run-on sentence. But
you generally use more commas than I do.
It's the Oxford comma, right? But anyway, I thought, you know, I could relate and understand Mike a little better from reading this paper. You know, I'm not sure I would recommend it to everybody, but this one, according to Google, can anybody guess how many citations there are? No, it's more than 0. No, it's less than 2. All right. Okay. There's another point that I really thought was telling because, you know, we started with one of Mike's first papers, and now, you know, if you zoom forward, this is one of Mike's more recent papers. This was an analysis of height across 5 million individuals organized by the GIANT Consortium. You know, and for those who are familiar with the history of the GIANT Consortium and actually with the history of meta-analysis of these genome-wide association studies, and we'll speak to that a little bit more later, you know, Mike was really instrumental in getting these consortia going. And it was a combination of really having the insight that it was important to combine data and to analyze it systematically and with the best methods across large numbers of individuals, but also having kind of the, I don't know, you know, the kindness, the patience to work with many different investigators. You know, I was in some of these very early meetings when we were talking about combining genome-wide association studies. I don't know where it's Mark McCarty here as well, but, you know, without naming names, we probably could remember, you know, Philippe Frogel and how interesting it was to convince him that this was a good thing to do. Sorry, that was a slip. I didn't. But the remarkable thing, when you take this kind of span of time, about 50 years from 1980 to now 2022, we went from about 400 individuals that were being assayed with this quality assay to measure HLA-D types, to now over 5 million individuals measured at over 1 million genetic variants. So from about 400 to something that's about 10 to the 12 or something like that. That's about 1 billion times more data. So that's actually a little faster than a doubling in the amount of data every couple of years. It's faster than Moore's law. It's faster than computer power has grown. And that has a lot of interesting implications. So it's not just that we have more data, but the kind of questions we're asking now are very, very different. And all along the way, they've been very different. Starting from originally trying to decide with just a tiny amount of data, was the trait genetic? Could I map it to a single chromosome? To later in genome-wide association studies, really pinpointing variation to specific genes, to having more and more detail and trying to understand what those genes do, what the variants do, to now stages where we're trying to integrate many different kinds of information. And Mike's been there along the way, and that points to kind of a curiosity and a passion for kind of lifelong learning and keep updating things. Most of us probably would have said, we've had a good ride so far, and we'd settle back and just let things be and watch from afar. But Mike's been in the thick of it for kind of quite a huge span. All right, so I'm going to take a detour. I know it's poor form, but just for context, I'll tell you a very short version of my story before I met Mike, and then we'll bring them back together and talk a little bit about some interesting things there. So this is the cartoon version of my background before 2001. And I think I actually met Mike in person at the end of 2000 at an American Society meeting, where we had that conversation that Mike recounted in a very nice, kind way. Another way to say it would probably be that, I wasn't very articulate and I couldn't explain what was going on, and it took 5 minutes to say, actually this might make sense. But, when I was a kid, I had a couple of things I really liked. I liked my ZX Spectrum, which is a very early computer. You could connect to your TV and you could read cassette tapes. And it turns out my parents wouldn't buy us video games, but they were happy to buy us programming books. At the time, they were basic programming books, not C++, where you could read up and code your own games. And so that's how I started programming. Eventually, I graduated to C++. And when I was in high school, I had a little software business that actually did pretty well. And with kind of the typical vision of youth, I remember when I was going to college, I said, I already have a successful software business. I know everything there is to know about software. I should learn about a different passion that I know nothing about. And so I decided to set aside that. And since I love biology and I had all these animal books, I said, I'm going to go study genetics. I know nothing about genetics. It would be very interesting. And so this is me and my siblings. And I ended up eventually at Oxford doing a PhD, working on the lab of Bill Cookson and trying out what at the time were high throughput technologies for genetic analysis. And high throughput at the time was a little better than Mike's polymorphic HLA-D assay, but it was basically strategies that allowed you to, say, measure a single genetic variant across 600 individuals at a time. And the basic strategy was, you make a really thin agarose gel, you make really small wells, so you have 600 wells in a gel, and you can load up a lot of individuals, and then you figure out how to score it efficiently. And if you can run a few of those gels a day, you can have hundreds of genetic markers. Now, this doesn't sound very high throughput now, but at the time, it was a major advance. And it turned out that the analysis tools that were available then really couldn't cope with the amount of data that we were generating in this kind of, pretty distant way. And I ended up, this was Oxford. I said, at some point, I said, our analysis tools can't cope. I think I could code something up. I used to run a software business. I know this stuff. And Bill actually was pretty sensible, and at some point he said, I have no idea if what you're doing is kosher. We need to get a second advisor who knows statistics and who knows the methodology and see if this is even correct. And so I ended up working with Lon, and Lon's basic strategy was, you don't know what you're talking about. Read this book and then come talk to me when we can have an informed conversation. And I have another picture, which was someone I ended up having a lot of conversations with in Oxford, and is actually Mike's younger academic sibling, I think, maybe twice removed. I think Dan was in between. Is that right? This is Eric Sobel, who also worked with Ken Lang. And at the time, he was in Oxford, and he knew everything there was to know about pedigree analysis. And he was also very happy to talk to anyone about it. Mostly, if you can tell that he has this wall full of Diet Coke cans, because he probably, was always very wired from the Coke. But it was great fun, and so I learned quite a bit. So one of the things, we ended up doing at Oxford, and this paper actually, this figure is from a paper written here in Michigan with Albert Smith, who's back there a few years later. But we were characterizing genetic variation across the genome and how patterns differ along chromosomes. And there's certain regions, some of you might recognize, this locus here where there's more structured patterns of variation is the HLA locus on chromosome 6. But there's other regions in the genome that look a little bit like that. In general, blue here indicates less structure. Those are regions at the ends of chromosomes where there's higher recombination. So there's a series of interesting patterns to study. And even though it's very esoteric now, at the time this was a highly debated topic, and you could get a Nature paper by characterizing it better. And again, I should go back to Mike's stories, but I have to tell this one because it was so funny at the time. So we wrote a paper showing that that the pattern of linkage disequilibrium in the genome was very different than predicted. And we sent it to Nature, and the reviewer said, we know this paper is wrong because it contradicts previous simulations. And so it got rejected from Nature, and then we sent it eventually to the American Journal of Human Genetics, and they loved it, and they published it they accepted it in a week, and they published it the following week, which was incredible. And then 6 months later, a different paper came out in Nature from Eric Lander and a couple of colleagues, and they said, consistent with previous smaller experiments, we now can confirm that there's much more linkage disequilibrium in the genome. And we're cited as one of the previous smaller experiments, except we have 100 times more data. So, anyways, you should always reject data based on simulations. Now, in the process of this, we were doing these linkage disequilibrium studies, and we had a strategy which was to study small families, and that made the analysis very simple and straightforward. However, there was a mistake. Someone didn't track the connections between the different families, and when we thought we had a series of small nuclear families, we ended up typing a large interconnected family with 24 different individuals. And this was kind of a huge problem for analysis tools at the time. It just was well beyond what could be done. After deciding, should we throw out the data, should we start from scratch, eventually we kind of had this intuition that there was an analogy between the repetitive calculations that you do in terms of linkage analysis. Usually you're cycling through an array of different possibilities. You could represent these in a kind of tree, and you could easily capture symmetries or patterns and really greatly reduce the amount of computation you had to do. And this was quite important at the time. On this example, it took a simple analysis from 16 minutes to a few seconds, and it could be extended and applied further. And so about that time, I was applying for a job, and I ended up talking with Mike. And Mike had this Center for Statistical Genetics here at the University of Michigan. Now, I guarantee I went and found the original 2000 August, it says 2020, but it's August 2000 webpage. This is my own typo. Sorry. But I found it from all the way back then, because if you put something on the web, it ends up on the web archive, and you can go dig it up. But if you go read the text now, the page has moved addresses. It's no longer here. Now it's a csg.sph. But the text is exactly the same. Nothing has changed. Mike hasn't taken his pen and said, this is a bit too wordy, could be phrased better. But I'll read a little bit, because there's a few interesting things to bring out there. The Center for Statistical Genetics is an interdisciplinary program, seeks to encourage research and training at the interface between human genetics and the mathematical sciences. Okay. I guess you could have just said Center for Statistical Genetics, interdisciplinary program at the interface of human genetics and mathematics. Save a few words, but it's okay. It's okay. But build a community of faculty and students, promote methodological research for genetic studies, foster use of innovative models and methods. And so, obviously, this is a very interesting place. And I remember when I interviewed here at Michigan, Mike had me have lunch with a bunch of students. And somehow, these students asked really tough questions. I don't know if they'd done a journal club in one of my papers or something, but they were deep into this stuff. I think it was Mike Epstein and Chun Li, and I can't remember who else was around back then. But I was like, wow, this is kind of a fun place. These guys know what they're talking about. If I was there with students like this, how much could one do? So I was probably pretty sold there, but what actually happened, and this is the punchline, but the way the story started, but I think it illustrates Mike's generosity, is a little different. Mike kept telling me about this Michigan football program, and if I came to Ann Arbor, I'd love Michigan football, and I should go to every game. I grew up outside the U.S. I'd never been to a football game. I probably watched a few minutes. I thought, this game doesn't make sense. Give me soccer any day. But I thought, this must be a real interesting, valuable experience. And Mike told me, before you accept, call the Michigan Athletic office, say, you've got a faculty offer from Michigan, you're a big football fan, and you would come if they would only give you season tickets. Because there's like a 7-year waiting list or something. And I said, well, these must be valuable commodities. I will call them and say, I'm a prospective faculty member, but I'm a huge football fan. I will only come if you give me these tickets. And they said, no, we don't do that anymore. So, well, I felt, what do I do? I guess, so I'd say, I have to call Mike up and say, Mike, I was about to accept, but, And Mike said, no problem, let me deal with it. So Mike, I either called the dean's office or called the athletic office, but the end result was he got the same answer, no. So then Mike probably had to figure out, what do I do? And he didn't know that. I was already impressed with the students and stuff. So he called me back and said, I tell you what, I have season tickets. I'll share them with you. And so for many years, we went to Michigan football games and sat in Mike's seat, So Very, very nice. Okay. Now, one of the great things about being here at Michigan and being in the biostat department, I inherited Mike's biostatistics 666 course. You know, there must be a backstory to how the name came about. But it was a really great course going through seminal papers in human genetics. I remember that the first time I taught it, you know, I taught with Jeff Long, and Jeff said, you know, we're going to teach the course, and then I have a strategy that will guarantee we're going to get great teaching evaluations. And so on teaching evaluation day, he said, you know, while the students fill the teaching evaluations, we're going to tell them we're going to go walk to South U and bring back some pizzas and pop, and then they'll be so happy they'll give us good evaluations. It doesn't work. It really doesn't work. But you know, eventually I found out that bribery was not the correct strategy, but it's possible to get, you know, pretty nice evaluations teaching a course where you know quite a bit about and the students are interested. Oh, actually, the strategy that does work, which is very antithetical to my personality, as some of you would know, but I tried using it every time, is to move the class from afternoons to 8 in the morning, because then you really only get the dedicated students. You know, genetics has a lot of people that are, I don't know, let's call them gadflies. They are interested in genetics, but not that much, and then you give them something they think is a beautiful, interesting result, and say, no, this is too complicated. Why are you making me try and follow this? But if you move the course to 8 in the morning, make the first class as hard as you can so most people drop it. The ones that stay give you amazing evaluations. And then you can teach at a normal, appropriate level. So, but, you know, so we were going through seminal papers. You know, one of the papers I always loved and connects with Mike's early focus on getting quality data and avoiding errors was the series of papers. You know, there's a paper by Mike and Nancy in '97 describing how to do relationship inference. There's a follow-up paper by Mike Epstein and Mike and Bill Duran, who I think is also here. Bill? And where they refined the method to work with a greater variety of relationships. But these papers are great to introduce a lot of interesting concepts, including hidden Markov models and how they can be used and how they can be fit and calculated. And so this was a great way to learn. You know, it was a great way to teach. It was a great way to find amazing students. You know, because, you know, one of the best ways to know that you've really learned something well is to kind of go and try and teach it, you know. And so this was kind of always a fun thing to say, you know, what are the parts of human genetics I want to learn next? You know, and you just add those papers into the syllabus and you figure out how to teach them. And you ask tough questions because you've selected the students to be students who are willing to tolerate the tough questions. But then by reading all their answers and so on, you really start learning something pretty deeply. And so, you know, obviously we relied on great papers, learned the fundamentals of genetics, and many of these were written by Mike. Now, in terms of taking Mike's advice on how to teach, I have another comment, which is Mike is a really good and clear writer. He writes great papers that are good to learn stuff from. He's also a great teacher. He puts a lot of work into preparation and so on. And so he makes a bunch of this stuff look very easy. And a few years later, I fell into this terrible trap. Teaching statistics to non-statisticians, you know, how hard could it be? And so this is one of my intro slides from this course, but Mike used to teach Biostat 501, which is a course with about 500 students. And most people, since they were probably had thought about it more deeply, tried very hard to avoid teaching this course. But there's, there were 3 or 4 people in the department, including Mike, who could teach it and get, straight A teaching evaluations, 5.0s and things like that. I think Bramar was one of the other ones, and she's also here today. And there was one year where I thought, if they can do it, I'm sure I can do it too. No. Yes. All right. So back to our story. Where we are at this point, when I got here to Michigan, in some ways, these were kind of the dark days of genetics. If you think about genetics as being kind of a great tool where people can now find genetic variants and try and learn about the biology of almost any trait, it wasn't like that then. It was more one of these stories when you go read about, I don't know, there's some family that started an NFL team in the 20s when football was only for hardcore aficionados, and people died playing games because it was so dangerous, and not that many people watched it. And now they're billionaires because they stuck with it for like 60 or 70 years. This is when people died and everything went wrong and nobody showed up to your games. You really had to love it and think that you're curious and want to learn interesting things. And this paper by Joel Hirschhorn kind of really makes the point in a very strong way. They looked in the early 2000s at 160 different genetic association signals that had been studied multiple times. And their conclusion was that only 6 of those consistently replicated, and that probably most of the others, if not all, were false positives. So this is not kind of a great place to be at. And I remember, many times because walking into Mike's office, and if I wanted to tease him a little bit, to say, Mike, the reason it's so hard to do genetics of diabetes is that you should have picked a trait that's not so environmental, And now, obviously, he's proved me wrong. He's done genetics of diabetes very well. And obviously, one of the early stages of moving on from these times, and coming up with strategies that really worked, a landmark at the beginning, that was this fusion study by Mike and many colleagues where they did a type 2 diabetes association study that was a genome-wide study. So instead of trying to do a candidate gene study, which basically goes along the lines of, I've collected these samples and these individuals, I want to study diabetes, let me ask smart people. I'll go ask David Ginsburg. David Ginsburg, what is the best gene to study for diabetes? And David will be very insightful, and he'll give me a good quality prediction. Study HNF4 alpha. But I won't take it just like that. I'll say, oh, now he's taking me HNF4 alpha. Let me pick one specific base pair at HNF4 alpha and guess that is the determining base pair. And I'll measure just that one and study it. And you could see that, because maybe not everybody's as smart as David, and picking one base out of a gene is probably a hard business. Most of these studies fail. Now, the difference here was to say that, because of better technology, we could study every gene and every available variant at the same time. And there it works really well once you have a large enough sample size. But the story I wanted to tell is some of the other interesting things that were happening around this time. Because what Mike shared was not just football tickets. Obviously, I appreciate the football tickets. But Mike was also very generous in sharing students. So Mike had always like a cadre of great students coming through, both because of his own grants and because of the Center for Statistical Genetics training program. And he was also always open to kind of trying to understand things from sort of from the basics. Think about, not just like, hey, this is how we always do it, but why do we do it that way? And there's lots of interesting things that come up if you pay attention. So for example, this is a paper that Andrew Skoll and Laura Scott, Mike and I wrote. And we were thinking about, what's the right way to analyze data from these genome-wide studies? And the standard practice, which basically was fine for the previous setup, where you pick one base pair or one genetic variant, and if you find something interesting, you say, let me do it again in a second study, see if it replicates, was to have a very hard focus on replication. Now, as you extend it to say, I'm testing 1 million variants, should I just pick the top one and see if it replicates in the second study? Maybe. Should I pick the top 10 and see if they replicate? Maybe. Those all could be plausible, but the default was to just say, we'll pick the top few, see if we could replicate it. It turned out that this was a terrible strategy if you care about getting the right answer and if you care about statistical power. So in the dark line here, you have, for a variety of settings, the power you would get if you took the optimal strategy, which is look at every variant in every available person. And then you have, in red, the power you get with the replication strategy, which is basically what everybody did by default because it was what was common before. And this is what you'd get if you took a joint analysis strategy, which is at least where you can look at all the available data and forget about trying to separate it into discovery and replication. And there were a series of problems like this, where I was very fortunate to be here with great students and with Mike, who was always happy to say, that sounds like an interesting problem. We should put, Andrew on it, or we should put Yoon on it, or we should put Christian Fuchsberger on it. Many, out of his trainees. And so, for example, another interesting thing in this paper was that we used imputed genotypes. I think it was the first paper that really tried to apply this methodology, and it was based on work by Yoon Lee, who was a student here, and Paul Sheet, who was a postdoc here at the time. I think he was on the training grant, although I'm not completely sure. And they investigated this idea that said, in families we know that DNA travels in big segments, but even in unrelated individuals, they're still distant relatives of each other. So there'll still be these shared segments. And if we could find these shared segments of chromosome, we could fill in the missing variants in between. And so the cartoon of what they're trying to do is to say, in our data, we have a small number of sites that we've examined with the array. There's now some sets of samples that have been genotyped in more detail, and they would actually apply a hidden Markov model, not so different from those that you would use for genotype relationship inference, to find these segments and fill in the missing data in each sample. And this turned out to both be quite useful because it increased power for genetic studies, so you'd increase the number of discoveries. But it also, by starting with a small number of variants in red and filling in all the other ones in blue, it made it really easy to compare results across studies. In the case of the fusion study, there were a series of diabetes studies done at about the same time. There was one done at the Broad with David Altshuler, who I think was here last week. There was one done in Oxford led by Mark McCarty, but they each used a different array. They used, at the time, an Affymetrix array, and Fusion used an Illumina array. That meant there was actually very little overlap. So if you wanted to compare results or to combine results across studies, and unless you did imputation or had some other convoluted strategies, and there were some convoluted alternative strategies at the time, you'd have a hard time doing that. And so this is an example actually this slide is out of order. It's not what I want to do. Anyway, I don't have this. So it's okay, it's fine. But this shows a follow-up analysis, and this shows that you could actually, this was a study the following year where we took all the data from these diabetes studies, a couple of other studies, and combining them, you take studies that basically had almost no discoveries, but now you could find 17 different loci that were influencing your traits of interest. In this case, cholesterol levels. Kristen actually is my wife. It was my wife at the time, so it's great to opportunity to work together. And then there were some interesting follow-up papers on the methodology to do this that eventually became standard. I will go back to the main event, but and now another example. Christian Fuchsberger, one of Mike's postdocs, eventually took this idea and figured out that the way to make imputation convenient and make sure that people use it all the time is to set up a web service where you could come, bring your data, you would compare it to the best available reference panels, return results, and then everybody could do it all the time. And now everybody does do it all the time. This slide is many years old, but when I checked this morning, the two large imputation servers that Christian helps run together with some other colleagues in the audience have now over 200 million imputed genomes, which is kind of incredible, and they have probably been used by almost 10,000 investigators from all over the world. Now, what I also think is not so obvious is that one part of it is have great students, have great people, give them time to pursue their ideas. But there's a lot of work that happened around here that was, for example, this is a new idea. Let's take the best available sequence data sets, put them behind an imputation server. Everybody else will come and use their data. It'll be simpler. But imagine being the first investigator that's saying, I'll let you put my data on this web server. What if it gets hacked? It wasn't such a popular idea when we were discussing it. But Mike really helped set up this thing that at the time was called the Haplotype Reference Consortium, where we found some sort of pioneer set of studies that were willing to share their data and work with the IRBs to find that it was okay, and work patiently through the process so that there was actually a data set to be compared against. So I think it's the genetics, the creativity, but spending time on these relationships that's also key. And so I wanted to maybe pause this chapter and say, look, these are many of the students that I worked with while I was at Michigan. And I think that Mike didn't just share his football tickets. He said, I'll share my students with you. And this was kind of a great, amazing thing. Obviously, some of these trainees are postdocs as well, so I don't mean to denigrate anybody. And this kind of idea of having a way to connect and work through people was valuable to me many, many times in my career. So when I think about my involvement in the HapMap project or the 1,000 Genomes Project, Sardinia, TopMed, the Precision Health Initiative here at Michigan, which was where this cartoon comes from. These were projects that required good ideas and good methodological approaches, but it required bringing together a lot of people. It required one thing that is actually one of my favorite memories of working with Mike, even though it's not good advice for anybody. But one of the reasons Mike and I worked well together is because our circadian clocks were so out of sync. And so when we had a grant deadline, and Mike is an amazing grant reader and an advisor, I could work until, 4 in the morning, and then I could hand it off, and at 4:30 in the morning, Mike would be up and reading it. And then when I got up, obviously, I'd have to be up at 8 to teach my class, but when I teach my class and it come check on the I had very detailed read-through, very detailed suggestions. It was great. But it's not only figuring out the grantsmanship, it's not only writing good grants and having good ideas, it's figuring out how to bring other people along. And Mike is, really knows everybody at Michigan. He knows people that I don't even know how he came to find them in the first place. when we did this MGI study, one of the key ideas there is that we wanted to make the consent really transparent. And so we worked to get the consent down to a single page. And we worked to then say, in addition to this single page that's got very declarative statements, I'll allow you to use a sample of my DNA, I'll allow you to use genetic research or so on. But something that fit in a page, and the IRB said, actually this covers all the points that are in the instructions. You don't need to use. Then we prepared a parallel little pamphlet that was a cartoon and illustrated to show all the different ways that this was valuable, this was risky. And it was interesting because the IRB was very forward-looking, and they thought, the worst thing that could happen is if we want to do something in the future and we haven't mentioned it earlier. And so if you read carefully on the side, you'll see that, they said, when we collect your DNA sample, we might use it to make stem cells that turn into other cells or turn into organisms, which maybe not so likely. It's something far beyond our plans, but they say, just in case, if someone doesn't want that to happen, maybe we shouldn't be banking their sample forever. But this was great, and it involved, for example, knowing this guy, Nick Stanek, who was an historian and an expert on IRBs and on research ethics, and he really had the vision for how we could pull all this off. Okay, so, some lessons that I've learned over the years working with Mike, and I think contrary to the circadian rhythm one, which I think is probably unhealthy, could be useful to some of you, is that, even one person, if you're paying attention, you have a good idea, it can make a difference. Obviously, part of the difference is the initial idea. Part of the hard work is then figuring out how do you take a good idea that, say, imputation is useful, that you should do joint analysis instead of meta-analysis, and figure out how do we change the practice of a field? Because there's a lot of papers out there that have great ideas, but many of them don't get very far. And you have to figure out the work of explaining it, writing not just in a statistical journal, writing in a journal where the people who are making what you think are the mistakes or who could do their analysis better will read it and understand. And, but anyway, but I think that's good, and I think it's still true. Even though the data sets are so big, there's incredible ideas all the time to be found out there. I think another thing that I think is really an important lesson is that the best students, the best postdocs and collaborators, we all have a tendency to say, let me imagine what I was like when I was younger or at an earlier stage of my career, and let me find a person just like me who knows what I knew then or something. But that's not quite the ideal situation. You really want people that know different things, because then you can do things that are beyond what you could do on your own, even if you just had more time. And so you can do really special when you find those great colleagues and collaborators. Sometimes you just got to drop things and go do what's fun and new. Mike was always very good about encouraging that. Obviously, keep learning. How do you go from starting analyzing HLA-D alleles, calculating allele frequencies, and so on, and end up now doing, functional studies with RNA sequencing and whole genome sequencing and so on. There's tons of great ideas out there. And the other thing is that actually the most valuable things are very simple. And that's an idea that probably takes a little longer to figure out, but I think sometimes there's great ideas that are very complicated, but very complicated ideas have a problem, which is it's very hard to then build upon them and combine them with other ideas. And so sometimes the simplest approaches and kind of their building blocks are great because you can remix and add them and build them into new things over time. And I think we're actually in an era where observing that in many ways. If you go and figure out, for example, how do these large language models work? And they do amazing things. But they're trained on predicting the next word, and they're basically a lot of matrix multiplication. And the way that matrix multiplication, the matrices got fit is because someone figured out, hey, here's a strategy to calculate derivatives on very large matrices. So you can calculate derivatives with respect to every element, evaluate them against how well they're helping predict your word. And because you have derivatives, you know how to fit the parameters. You can just move them along in the correct direction on about the correct scale. And you just do it again and again. And you've got ChatGPT, as opposed to, previous language models, which were much more sophisticated, tried to build a lot more structure, but obviously couldn't scale, couldn't to be fit or understood on large enough data. So, okay. So I don't know if I need the next little bit, but I think I still have another 5-10 minutes by my count. If someone wants to kick me out, it's okay. There'll be no hurt feelings. I know. But I thought, I was worried that Mike was thinking about retiring. And so I really wanted to just finish by making the point that, if you believe that these are worthwhile goals for human genetics, to understand disease, to predict disease, we're kind of in a golden era. Like the sort of things that genetics can do are amazing, and well beyond what we could have imagined, when I started, and certainly when I imagine, unless Mike was, I don't know, the Isaac Asimov of genetics, and he's like predicting the future, I don't think he could have imagined it either. Just last week, we, some of my colleagues had got approved for a gene therapy for total deafness, for congenital total deafness. It's for one specific kind of congenital deafness, but it was something that, in the early 90s, gene therapy was an idea out there, but no one knew how to make it work. No one knew how to make it work even in model organisms. And the fact that now this is not even the first gene therapy, there's other approved ones, it's kind of amazing. And you need to have really great genetic data, understanding of the genetics and the biology to figure out places where these things can work. We're also now really good at understanding and discovering new biology and new possibilities. I was going to show a couple of examples. So I've been at Regeneron about 8 years, and one of the first kind of analysis we did is this very simple analysis. Regeneron has a medicine that works by blocking IL-4 and IL-13, and it's very effective at reducing asthma adverse events in people with severe asthma. And we just said, what if we could build a little genetic risk score only using genetic variants near IL-4 and IL-13 for asthma? And you can build a great score, as illustrated there. But now that you know, hey, the drug works by blocking IL-4, it's effective for asthma. Now you have a genetic score, recapitulates the drug. Now you can test the score against other indications. And so when we were doing these analyses, you could start with asthma, you could try it on atopic dermatitis, and you say, look, this probably will work for atopic dermatitis. You can try it on nasal polyps, and you can say, oh, it probably will work on nasal polyps. You could try eosinophilic esophagitis, so inflammation in your esophagus, probably will work there. Prurigo nodularis, maybe. It's a sample size, it's harder to read that one. And probably won't work for rheumatoid arthritis or Crohn's disease. And these were all indications of interest, and actually now, they were not 8 years ago, but they are now. These are all approved indications for this particular drug. So it turns out the genetics could give good advice. And so one of the fun moments I remember having early on, we were having a meeting in the boardroom with the guys who ran clinical development, and they said, okay, why don't you take those predictions, write them on the board on the side, and you don't know, but we know, we just got this morning results for a phase 2 trial in one of these indications. Let's see if you got them right. And so happily they were right. Otherwise, maybe I'd be happy back here at Michigan instead. But it was fun. And I also wanted to show another little vignette that has a Michigan connection. Chris is a student who worked here with Matt Sampson and with Hayun, really smart guy. And he was interested in this problem of trying to predict whether a genetic variant in a protein sequence is likely to damage the protein or is likely to be not so damaging, which is very interesting for genetic association studies. If you're combining information on lots of very rare non-synonymous variants, you want to say, I want to combine only the ones that are likely to damage the protein. I want to set aside the ones that probably are harmless. And there's a few kind of observations that turn out to be, at least to me, were completely unexpected and really remarkable. So the first one is, what happens if you just say, try and use a language model to read protein sequences, to take all the proteins that have been sequenced, there's about 100 million of them, train a language model on those, and say, now I can take any potential protein sequence, I can mask one of the amino acids and try and fill it back. And I can ask questions like, well, when I fill it back, what is predicted? What do I actually see? Is this like a unlikely prediction given all the context of the entire protein? That simple model, with basically no functional data, no knowledge of structure, all it does is it's read all the proteins across the tree of life, and it's now trying to fill in little gaps, just like a large language model would fill in words and text. It's better than Polyphen, it's better than SIFT, it's better than all the previous kind of expert systems designed to solve this. And so that was kind of interesting. The other kind of thing is that if you can find a way to combine the large language model, which is just basically pattern recognition across all these 100 million proteins, with data that you have to guide it on what is actually deleterious or not, you can even further improve it. And so this is just a little slide to kind of illustrate the challenge and say, this is not a new problem. If you go back to 1974, Grantham wrote a paper in Science saying, I looked at all the different possible amino acids and I paired them up and I measured a series of characteristics. Were they positively or negatively charged? Were they large or small? What fraction of carbons and nitrogen did they have in their side chains? And it came up with this amino acid substitution matrix. And it's still, a pretty decent starting point. And you can say, hey, these are the amino acids that are closest or furthest based on size, polarity, and some kind of composition. But you want to figure out, can you gradually do better than that? And obviously, there's many improvements on this problem. And what we're trying to do is to take any missense variant and say, is it more like a truncation and frame shift, like a peel-off removes protein function? Is it more like something that doesn't alter? And what we ended up doing, this was Chris and Hayun's idea, was to say, well, we have a ton of proteomic data. We have, at the time, we had data on about 50,000 individuals where we'd measured protein levels using antibody assays. And we also knew the exact sequence because we'd sequenced those individuals. And we had the hypothesis, well, if the protein sequence has been altered in a meaningful way, the antibody assay results will vary. And so we can focus on the missense variants that lead to the largest changes in antibody readouts and try and say, hey, here are examples of missense variants that we think are good and bad, and combine the large language model with a classifier trained on that and now apply it to completely new proteins. And it works really well. It's a busy slide, but this tries to show if you take, on the one end, variants that are known to be truncation variants. There's very few of these in this particular gene. This is a gene called 6.1. It's known to be associated with hearing loss. If you take all the non-synonymous variants, the effect overall is pretty small. And then you have different strategies. You can combine all the missense with all the peel-offs, and actually the missense overwhelm the peel-offs. Or you can try and find different ways to say, which peel-offs are likely to be damaging. And as you pick them better, you see you get actually quite decent effect sizes, and the statistical significance greatly increases. You went from a 0.01 to 10 to the minus 11, 0.003 at the other end if you took all. And obviously this is one example, but you can systematically evaluate this across all genes where you have some validation of the link between the phenotype and the disease. And we've done this across a few hundred. It seems to work. And the other thing is that it actually does, for example, better than, say, alpha missense and some other models out there that are kind of trained on similar tasks. Okay. You guys are about to be rid of me. Dinner, beer, whatever. But I have just a couple more things to say. So first, Kristen and the kids who couldn't be here today say, thank you and congratulations, Mike. And Mike, I never knew him in this stage in his life. I knew Mike when he went to the Galapagos, when he went to Machu Picchu, when he went to Bolzano. But he always used to say, I never used to take vacations, but then someone told me that it was worth taking a vacation. It was so great. Now I do it every year. I make sure me and Betsy go somewhere interesting regularly. I'm glad, he's taking vacations now. I don't think it was necessary for his success to spend 20 years not taking vacations. I definitely try not to imitate that. So this was us in Rome, as you can tell. The other little side comment I wanted to make, in starting a journey and not knowing where you'll end up, I think, this I'll share my tickets with you is like, this kind of bumbling way, I should take those tickets because they might be valuable. The fact there's a 7-year waiting list and so on. And then, getting really hooked on Michigan football and ending up watching, the Wolverines win the national championship a couple years back with the kids. And so, that's really been kind of a terrific, terrific thing. And I'm really appreciative of Mike for that opportunity. And, I guess I have not been in Ann Arbor enough recently, so I hope my final slide does not strike the wrong tones. But if it does, my apologies, but it is genuine. So it says, who has it better than us? Nobody. And it says, because of you, Mike, we here really are the champions and the best. So thank you very much.